Alright, so today on Shey's Microbiology, we are diving into something that really sits at the heart of bacteria identification: biochemical tests. These tests may look simple, but they give us some of the most reliable clues when we are trying to figure out who a bacterium really is. Let's walk through everything slowly and clearly. So, biochemical test basically shows us how bacteria behave metabolically. Every organism has a kind of metabolic fingerprint, and these tests help us read it. When I say biochemical tests, I'm talking about things like how bacteria use sugars, whether they produce certain enzymes, how they break down substrates, or whether they release gas, acids, pigments, and so on. It's like asking the bacteria, hey, how do you survive? What do you eat? What enzymes do you produce? And based on their answers, we identify them. Now, this part is key. Biochemical tests matter because many bacteria look the same under the microscope. Think about it. Two organisms can both be gram-negative rods, but one could be E. coli and another could be Klebsiella and another Salmonella. Different organisms, different behaviors, different clinical implications. So biochemical tests help us differentiate these look-alike bacteria. They bring clarity when gram stain and colony appearance are not enough. Okay, let's start with enzyme tests, some of the quickest and most useful ones. The catalase test. This test checks whether the bacterium produces the enzyme catalase, which breaks down hydrogen peroxide into water and oxygen. If you see bubbles, boom, catalase positive. This simple reaction helps us separate staphylococcus, which is catalase positive, from streptococcus, which is catalase negative. The next, we have the coagulase test. This one is mainly used to distinguish Staphylococcus aureus, which is coagulase positive, from the coagulase negative staph species. Staphylococcus aureus produces the coagulase enzyme that clots plasma, and that clot is basically saying, yes, I'm aureus. Oxidase test. This checks whether bacteria have cytochrome C oxidase. It quickly separates groups like Pseudomonas from Enterobacteriaceae. These enzymes tests are very fast and very diagnostic. Now, carbohydrate fermentation tests. One of the classics. Here we are checking if bacteria can ferment sugars like glucose, lactose, sucrose, mannitol. If they ferment the sugar, they usually produce acid and sometimes gas. So you see color changes or gas bubbles. For example, E. coli ferments lactose, Salmonella does not. This single point alone can help you distinguish major pathogens. If you're dealing with Enterobacteriaceae, trust me, IMViC is your best friend. IMViC stands for indole, methylred, Voges pascaro, citrate. These four tests together can clearly separate bacteria that otherwise look similar. Let's look at some other important tests. The Urease test. Some bacteria break down urea into ammonia. If urease is present, the medium becomes alkaline and turns pink. Proteus species are famously urease positive. TSI, triple sugar ion test. This one checks multiple things at once: glucose, lactose, sucrose fermentation, gas production, hydrogen sulfide formation. If the bud turns black, hydrogen sulfide is present. If cracks or bubbles form, gas is present. If the slant and bud change colors differently, different sugars are being used. TSI is likely a summary card for Enterobacteriaceae. Now, let me say this clearly. No single biochemical test gives you the full identity. We combine gram stain, colony morphology, enzyme test, sugar test, special test. And when you put all these results together, a clear bacterial profile emerges. This is how we confidently say this is Escherichia coli, this is Pseudomonas, this is Staphylococcus aureus. It's a whole puzzle, and biochemical tests provide most of the pieces. So yeah, that's the flow of biochemical testing in bacteriology. It's practical, it's clear, and it's honestly one of the most satisfying parts of lab microbiology, because the results tell real stories about bacterial metabolism.